This module is a continuation of our discussion of translation. We've already seen initiation. I'll briefly remind you of the last steps of it just to pick up at a reasonable place. Then we'll look at elongation and termination. In the synthesis of any polymer, we can envision the polymer synthesis or polymerization as these three steps: initiation, elongation, and termination. What you should get from this presentation and the previous one on initiation is that it is possible to dissect each of these steps by assembling components of the cell that do one and then the next and then the next process. So watch for that as we go through this description and review the initiation voiceover PowerPoint to see it in that one as well. So here's this reminder. In this context, it was possible to isolate the F-met-tRNA sub-F-met, that is the formal methionine tRNA, with its anticodon to AUG, which is the code word for methionine. It was possible, you may recall, to, in a prior experiment, actually assemble a 30S or small E. coli ribosomal subunit with a message using the appropriate initiation factors, which could be isolated from the cytosol of a cell. Remember, all this is based on fractionating cells and finding the parts that are required to do each step of translation. So it was possible to actually isolate large amounts of this thing, which has no name, which is the mRNA 30S subunit initiation factors complex. And then if you just added, as it turned out, GTP and the F-met-tRNA, they would associate and they would produce this thing called the initiation complex. So that's pretty much where we left off. And this step requires the F-met-tRNA sub-F-met, the formal methionine. It would not use any of the other tRNAs with their amino acids. It would not use the normal methionine tRNA sub-methionine. It had to be this initiator tRNA with its formal methionine. This is what happens in bacteria, worked out in E. coli. And then the next step is the completion of the ribosome and ultimately then the elongation process. So the next step was simply to throw in a 50S or a bunch of large subunits in with the initiation complex, which, remember, again, could be synthesized in its entirety. So let's just back up here. What you are looking at now is the initiation complex isolated from a prior experiment. And then from another test tube, we throw in 50S subunits, and lo and behold, they associate with each other, and one can follow the fate of various components. Specifically, the GTP was hydrolyzed to GDP and inorganic phosphate, and the initiation factors have come off. This complex has no name as well, although some people refer to this as the initiation complex. If the prior one was the initiation complex, then this one has no name. So now we're ready to look at elongation. So here's that structure again with the F-met tRNA sub-F in place, shown in red here. And the whole thing is assembled, and now we want to know what's the next step. And remember, we can isolate this complex, no name complex, from test tubes by doing the appropriate additions one at a time. So now we can add 20 different amino acids, which are going to be required, of course, to make any polypeptide. We would add a free energy source on the grounds that this polymerization will require energy, and so in fact ATP was added, or something that would produce ATP was added. And in order for the event you just saw, for elongation to take place, the cytosol of the cell had to be added, and eventually, as you can imagine, we could isolate things from the cytosol specifically that would be required for elongation, specifically a number of elongation factors. So if you provided the ATP, and you provided the 20 amino acids, and you provided some of the cytosolic factors that would be necessary to do elongation, you would get it in a test tube. This is, again, cell-free protein synthesis or in vitro protein synthesis. Again, in vitro means in glass, and it's meant to say outside of the cell. I don't know if I said this in class or at any other time, but when this sort of experiment was done for the first time, it garnered newspaper headlines that said, "Life in a test tube." Now, that might have been hyperbole, but it was still very dramatic. So the cytosol components that were required for elongation included a number of elongation factors. It included more GTP, and it included an enzyme called peptidyl transferase. We'll talk about that, as well as an enzyme called translocase. We'll also talk about that enzyme. So here is the first step of elongation. As you might expect, the second amino acid that is to be put into this newly growing chain has to be attached to its amino acid, and so here we see amino acid 2 on its tRNA, and adding this to the complex required GTP, as well as elongation factors. We recognize on the ribosome that is about to begin translation, or that is in fact going to do elongation, 3 sites. They're basically binding sites. We have the A site, which you see on the right, and that's the amino acid entry site. As you might guess, the amino acid 2 with its RNA, tRNA, is going to enter at that site. The P site stands for peptidyl site, and you will see why that's called the peptidyl site in just a moment. And the E site is the exit site. Again, you will see why that would be called the exit site in just a moment as we follow what happens. So if you add the appropriate elongation factors, these are, again, proteins that one could isolate from the cytosol by doing protein purifications from cytosol and asking which proteins are responsible for the next step. So you have elongation factors. Here's one elongation factor. And in order for everything to happen that you're about to see, GTP also had to be present. So GTP actually bound to that elongation factor as part of its function. And as the second amino acid tRNA enters the A site, that GTP is hydrolyzed to GDP and inorganic phosphate. Well, if you keep consuming GDP, you have to start regenerating GTP. So another elongation factor, a different one, is actually an enzyme that catalyzes the phosphorylation of GDP again. So you keep regenerating GTP. It's interesting, this elongation factor is not an intimate part of translation, but it is a necessary component to make sure there is always enough GTP to support elongation. The next step, now that you have 2 amino acid tRNAs, one in the P site, one in the A site, is peptide bond formation. And that is the result of the action of peptidyl transferase. And as you can see, a peptide bond, maybe you can't see that, it's a little small, right? But there is a peptide bond that is formed between amino acid 2 and formal methionine, and the amino acid 2 is at this point still attached to its tRNA. So what has happened is the FMET has been transferred from its tRNA to the second amino acid to produce what we see here. Now it says here that the peptidyl transferase is actually a ribosome. Again, a ribosome is simply an RNA enzyme. And you will recall that there is a ribosome activity that can self-splice introns in organelles. Well, this is a ribosome activity that is present in E. coli ribosomes, so it's a natural part of whole cells, not just organelles. And this one is responsible. This is actually a component of the ribosomal RNA, the small ribosomal RNA, 16S ribosomal RNA, that would be present, or rather, I think it's actually one of the larger ribosomal RNAs. Someone can tell me after they've looked it up. But in any case, it is one of the larger ones in the large subunit. So it's actually an RNA enzyme, and apparently none of the proteins in this large ribosome complex seems to have much to do with the creation of that peptide bond. The rRNA that does this spans the P and the A site apparently, so it's a function, it's not tied to one site only. That's not the end of elongation. Now that we have a dipeptide formed, at least at the beginning, we need to free up the A site, put the next codon into the A site so the third amino acid can get in. That requires GTP. and it is catalyzed by, this time, a protein that is a component of the ribosome called a translocase, and it catalyzes translocation, which simply means the movement of the ribosome along the message. And so you saw that happen in the animation. Again, this requires GTP, which is hydrolyzed in the process. This is one full cycle of elongation, and note that it has actually cost the equivalent of 3 ATPs, 3 nucleotide triphosphates. What are these 3 nucleotide triphosphates? Now, be careful when you think about this. There are several GTPs involved in first steps, and you'll see in last steps. These don't really add in very much, but to add each amino acid to a growing chain will cost 3 nucleotide triphosphates, the equivalent of 3 ATPs, and here they are. You need an ATP to stick each amino acid onto its tRNA, catalyzed by amino-acyl tRNA synthase, right? You need a GTP to bring each amino-acyl tRNA to the A site when the A site is finally freed up after a translocation. And then, as you might imagine, movement, right, motility, translocation, even though it's a very small movement, the movement of this big ribosome along the messenger RNA also requires the hydrolysis of a nucleotide triphosphate, and again, as you just saw, that is another GTP. Making a polypeptide is perhaps one of the most energy costly things a cell can do. It's a very energy costly polymerization. Elongation continued. Here we have the next amino acid 3, bound to its tRNA. Again, it requires that elongation factor bound to GTP, and as the third amino acid tRNA enters this complex now after the translocation, the GTP is hydrolyzed to GTP and inorganic phosphate again. We can throw in that second elongation factor that regenerates the GTP, just to remind you that you have to do that if you want the process to continue, right? Now we had this original tRNA, and if we're looking at the beginning of elongation, this would be the tRNA for F-methionine. It was sitting in the E site, and as you saw, it just left that ribosome. And that's going to be necessary so that you can begin the next round of elongation. You will, doesn't show, let me back up here. It doesn't show it in this slide, but the next step, of course, will be to transfer the dipeptide, that is AA2-FMet, to AA3. And now that's not shown, but if you want to draw this for yourself to see what's going on, you can. There is a transfer of this dipeptide from the transfer RNA in the P site now to amino acid 3 to make another peptide bond. And of course, then the cycles continue. But I wanted to show you what happens to formal methionine. There is an enzyme called formylase that catalyzes the removal of the formal group from methionine. This happens to all polypeptides that E. coli makes, or that bacteria make. Remember I think I told you, that about 40% of E. coli proteins actually begin with methionine, which is what led investigators to suggest that methionine might be the first amino acid laid down for every polypeptide, and as it turns out, that's true. It's laid down as formal methionine, but the formal group is removed, and that's removed by this enzyme called formylase. What about the 60% of polypeptides that don't even have methionine? Well, there is a proteolytic enzyme, an enzyme that can catalyze amino acids from polypeptides, and that removes the methionine. Such enzymes function to remove either the methionine or sometimes even the methionine and a few other amino acids, and proteases do that. And that brings us to the end of the module on elongation. Look for the module on termination and a few comments on eukaryotic variations on translation as well.